Welcome to that universal. Quickly, this is schedule A-level CIE biology. So I want to quickly look at animal transport. I wish you the best in exam this morning too. So get to writing materials. Let's solve some questions. The first one says, see there. Describe the role of the sinotral node and the atrioventricular node (AVN) in the initiation and control of cardiac cycle. I want you to pause the video and try it. Okay. So sinoatrial node is called a pacemaker. Right? It's found at the right atrium. So the function is that it sends out, it generates and sends out waves of excitation, which spreads across atrial wall. In fact, it generates the electrical impulse, which causes the heart muscles to contract. Okay, so atrioventricular node causes a delay, okay, in the passage of the electrical impulse into the ventricular wall. Why? So that the ventricles and the atria will not contract at the same time. Okay, to allow atria to empty and ventricles to fill with blood. All right, so look at it, guys, quickly. So you see, act as pacemaker for sound, initiate heartbeat. That pacemaker is any of this is symptom. Okay, releases waves of excitation, action potential, which spread across atrial wall. Okay, so AVN short delay passes the impulse waves of excitation to porcine tissue. Yeah, it does this, gives to porcine tissue. So atria contract before ventricle, allows ventricles to fill, allow atria to empty completely. So atria have empty or contracted before ventricle contraction begins. So atria and ventricles don't contract at the same time. So that is AVN for you. 4 marks. The porcine fibers pass down the septum and extend to the cardiac muscles at the base of the heart. Explain why it is important that the porcine fibers extend to the base of the heart. Yeah, it is important because, anyway, pause the video. But you know it's important because they carry the impulse from, the base of the heart to the upper part of the heart. So the impulse spreads from the base of the heart so that the wall of the ventricular wall can contract from bottom up. Okay, so if you check here, watch this. Okay, it should contract from bottom up. Yeah, I'm just sketching a heart. Yeah, just sketching. Yes, so just to show you. Yeah, just to show you something. Yeah, so you see that. All right, if you look at this, you see that it is very important that this extend to the heart, because it allows the ventricle to contract from the bottom up in order to pump blood. This is left ventricle is going to pump blood into the aorta, and then the right ventricle pump blood into the pulmonary vein, pulmonary artery. So pulmonary artery and aorta, they are the upper part of the heart. So the left ventricle has to start contracting from the bottom in order to squeeze blood, pump blood into these arteries. Okay, so just to make up on this our sketch, so it will make more sense. Okay, though we're not focusing on see. So just to make up, So, but guess what? It has to contract from the bottom in order to pump blood into the atria, aorta, and pulmonary vein. Okay, because they are the upper part of the heart. Look at it. So contraction of ventricular begins. You can see where it begins at the base of the ventricles. Both ventricles contract at the same time. Yeah, they contract at the same time. But blood is forced upward. That's it. Okay, they are forced up through seminal capillary. I did that. So ventricle empty or pump most of the blood. Okay, so impulses of excitation travels upward, so that blood is pumped up Okay. All right, quickly. So in mammals, arteries branch to form smaller blood vessels called arterioles. Arterioles branch to form capillaries that supply blood to tissues. Explain the ways in which the structure of an artery is adapted to its function. So, pause the video and try it. How is an artery adapted to its function? Okay, remember that arteries have thicker walls, right? It has just like vein, but it has thicker tunica externa. Externa is the outermost wall. Okay, it has thicker tunica media, which is the middle layer. Okay, so sorry. So why does it have thicker walls? So the question is, what structures are present in these walls, these layers? What structures and what is their function? That's what matters most. What is their function? So if you look at tunica externa, for example, that's the outermost layer. The outermost layer has collagen fiber and elastic fiber. So collagen fiber and elastic fiber, they play an important role together. This is tunica. This is tunica externa, tunica media, tunica intima. So here has collagen fiber and elastic fiber. So they prevent the artery from bursting or rupturing due to high pressure. So here we have elastic fiber too. Elastic fiber, we have a lot of elastic fiber and of course collagen. So elastic fiber stretches, okay, and of course that prevents the artery from bursting, but it recoils to maintain the pressure. It maintains the pressure, so we have more of it to maintain the pressure, okay, apart from bursting. The smooth muscle controls blood flow, the volume of blood flow. Smooth muscle is found here, okay, together with elastic fiber, together with collagen, but collagen and elastic fiber are found here, okay. Now here we have the tunica intima, the endothelium. The endothelium smoothens out, reduces the friction between the blood inside the lumen here and the wall of artery. Okay, it also protects the other layers. So you have to mention every part of it. So you see, thick wall tunica media, so withstand blood pressure to prevent bursting. Endothelium, okay, has smooth little friction to blood flow, okay, ease flow. Elastic fiber stretches to allow surges in blood or recoils to maintain blood pressure. Of course, smooth muscles contract to maintain or regulate control of blood flow. Collagen fiber, so you have to mention the structures that are present in these layers and what they actually do to help artery carry out its function. Fig 4.1 shows transmission electromicrograph of cross section through an artery and capillary. Okay, so this is an artery, arterial and capillary. This is capillary. Okay, one cell thick. All right, they said identify the cells inside the lumen of the arteries in Fig 4.2 and state one reason for your identification. So the cells into the lumen of arteries. Okay, this is lumen arteries. Can you see these cells? Pause the video and identify that. What cells are these? Okay. The cells are red blood cells. How do I know? They have biconcave disc shape. Okay, yeah, and they have no nucleus, no organelles. They have homogeneous or uniform cytoplasm. Okay, they always are like asking this question. So biconcave shape, no nucleus. Okay, uniform. All right, so you can see that. All right, describe the differences between the arteries and the capillary that are visible in Fig 4.1. So let's go back. So what are the differences between the this is a difference between the arteries and what? Okay, and capillary. Yes. So this is capillary. That's the artery. You can see that. Pause the video. The artery has thicker wall, right? This has thinner wall. If you check, you see that the endothelium of artery is thicker, bigger, wider. Why this is thinner? Okay, the endothelium. If you check also, you see that here the lumen is bigger. Can you see it can contain the red blood cell, they can flow seamlessly in the arteriole, but here the lumen is smaller, so red blood cell is squeezing through. Can you see the shape of red blood cell? It's squeezing through, okay, the capillary, okay. Anyway, know that the artery has other, right, elastic, yes, elastic fiber, smooth muscle, but it, elastic fiber, and of course, okay, so you can see that. Let's see, yeah, so what I'm asking says, right, thicker wall, more than one layer of cells, tunica intima. So that is for artery. More cells forming perimeter, wider vessel, wider lumen. Yeah. So nucleus present in wall only in arterial. Cells lining endothelial are thicker. The lumen smaller. Okay. More red blood cells. Yeah. So because it has more lumen. Okay. All right. Let's look at it. Fig 4.2 shows a capillary network in mammalian tissue. It shows indicate the direction of blood flow of fluids. Let's look at it. Good. They say capillaries have a role in the formation of tissue fluid. Explain how tissue fluid is formed in capillary network. So how is it formed? How is tissue fluid formed? So this is capillary network. Pause the video and try it. Okay. This capillary network, we have the arterial end, the one that's close to the artery. We have the venous end close to the vein. So it is formed at the arterial end. Okay, how it is formed is that because at the arterial end, the hydrostatic pressure, okay, is greater in the blood, in the capillary compared to tissue fluid. So that forces tissue fluid. So that forces blood plasma to leak out through the endothelial pores of capillary. So they are forced out here. Is that okay? Due to high hydrostatic pressure. Do you get it? And they are forced out, okay, but the only thing that can go out, there are small proteins, and other substances that are small, all right, like glucose, amino acid, water, mineral ions. They are the ones that are forced out through the endothelial cords. So it's forced by due to hydrostatic pressure gradient, and these small molecules is what actually common. So we call the hydrostatic pressure of the blood at start of capillary here is greater, so causing leakage of movement, pushing out plasma. So these are the things that move out. Okay, so large plasma protein not filtered out, even red blood cell, because they are too big. All right, they are too big to pass through. Okay. All right. Okay, the vessel level X, if you carry excess tissue fluid back into the circulatory system, name the fluid inside the vessels labeled X and state one way which its composition differs from blood. Okay, so if you go back to that, this X, consider this X is called the lymph. The fluid is called lymph. The fluid is lymph. The vessel is lymphatic vessel. Okay, all right. So that is it. So some of these tissue fluid, they're supposed to return back to the capillary at the venous end, but some of them drain away into this blind-ended vessel called the lymphatic vessel. So this is lymph. So how is it different from, the blood? I think that's what asking us. Okay, difference. State one difference in which it's different from the blood. It does not have large plasma proteins, like albumin. It doesn't have red blood cells. Okay, so it has less oxygen, more CO2, less glucose. Look at it. All right, so it's the lymphatic lymph. No name. It doesn't have large protein. Okay, so you see, it doesn't have. It has no or little less oxygen, glucose, but it has high concentration, more carbon dioxide. It has high concentration of more fat, lipid, lipoproteins. Okay. All right. Okay. If you give 1 mark. All right. Difference between plasma. All right. Good. So fig 3.1 is a photomicrograph of a section through two different types of blood vessels, X and Y. Okay. So X and Y. This is beautiful, amazing. Okay, it said name the two types of blood vessels shown by X and Y. Okay, yeah, name the two types. Of course, this is artery. We'll pause the video and answer some of this question. This is vein. What's different between them? Artery has thicker wall. You can see it has thicker tunica media. This has thinner wall. Okay, vein. Artery has it has regular cross section, circular. This has irregular cross section. You can see. Artery has small lumen. This has large lumen. Okay, relatively wide lumen, if you want to say that. Okay, yeah. So based on what we have here, so let's look at, yeah. So A is artery, X is artery, Y is vein. One mark, just one mark. Okay, this is the same question. All right. This is II. State the reasons for identification of the type of blood vessel shown. We have mentioned it. Pause video and try it. Okay. Yeah. Of course, this is vein because it has thinner wall. This has thicker wall. Small lumen, larger lumen. Okay. It has regular, circular cross section. This has irregular cross section. You can see that here. Cross section not regular, not defined shape. That for this. Tunica intima smooth. See here, it has a smooth tunica intima, but artery has a, rough or wavy. That's a tunica intima of artery is wavy. That of vein is smooth. Okay. Wider lumen diameter relative to wall thickness. So this has wider lumen compared to this. Tunica externa adventitial than the tunica media. The external is thicker than tunica media for this. External is thicker than the media. Middle one. For vein, for a vein. Alright. Tissue fluid and lymph are formed when blood arrives in the capillary networks of blood vessels. Explain why tissue fluid is more similar to blood plasma than it is to blood. So try to solve this question. Why is tissue fluid similar to blood plasma? Okay, maybe they contain the same composition. Okay, they have the same composition, the same glucose, amino acid, water, mineral ions. Okay, and then maybe because they have the same viscosity, right? And maybe because the blood, if you look at tissue fluid and plasma, they have the same color. All right, both of them don't have blood. Look at it. They do not have red blood cell. You can see both of them are because red blood cells are too large to pass through endothelial pores. Because they say we should explain it. That's what they explained here. Tissue fluid and blood plasma similar viscosity, similar in terms of color. All right, they don't have red blood cell, large plasma protein because it cannot pass through the endothelial pores. Okay. All right. So look at, explain why tissue fluid formed after blood arrives in the capillary network has a higher concentration of amino acid than newly formed lymph draining away from the network. Pause this video and try it. Why is it that tissue fluid that arrives have higher amino acid than the lymph? Okay. Look at it. Let me just help you with this. Okay, look at that picture. Just want to help you. Right? I just want to help you. Come. Where is it? Look at it. The tissue fluid that just arrives here has higher amino acid than the lymph that's draining away. Okay. The reason is because the amino acid is taken up by the cells. As you can see, the cells take up amino acid, and what do they use it for? They use it to make proteins. So as the tissue fluid flows around the tissue, back the tissue, the tissue or the cells take up amino acid and use it to make protein. So by the time the tissue fluid drains into lymphatic vessel to form lymph, most of the amino acid have been taken by the cells, tissues, to make protein. You got that? Okay, so that is the reason, guys. Okay, let's get back to where we are. Okay, I needed that diagram to explain that for you. Okay, so yeah, I think okay, this is where we are. You can see that. So most cells taken up or transported into blood, okay, used to synthesize so amino acids to make proteins like enzymes. Okay, just one mark. Fig 3.1 is a transmission electron micrograph showing red blood cells in capillary of healthy adults. Yeah, this is red blood cell. Explain how the cells in the capillary shown in figure 3.1 can be identified as red blood cells. So pause the video and try. How is it identified as red blood cell? It's simple, but try it. You can see that they have no nucleus, okay, uniform cytoplasm, okay, and you can see that their size is the same as the lumen of the capillary, so they are squeezing through. So the size of capillary is 7 micrometer, that's the lumen of capillary, and the size of red blood cell is 7 micrometers. So these are the things they always like asking this question. No nucleus organelle, right? Cytoplasm is homogeneous. Please don't forget it. About same size or width as lumen of capillary. That's what they're squeezing through. Flexible, variety of shapes, regular. Cells about. See that? This question has come. It comes out almost all the time. Pay attention to it. Alright. Capillaries are surrounded by tissue fluid. Outline the ways in which the composition of tissue fluid differs from red blood cell. So pause the video and try. How is tissue fluid different from red blood cell? From blood, sorry. So tissue fluid does not have red blood cell. Okay, blood has red blood cell, and you know the reason, because red blood cells are too large to pass through the endothelial pores. Tissue fluid doesn't have large plasma proteins. Blood has. Okay, tissue fluid has lower concentrations of oxygen, amino acid, but yeah, it has higher concentrations of oxygen, yes, amino acid, you get, but it has lower concentrations of CO2. Okay, it also has high concentration of glucose compared to blood has higher concentration. What I'm trying to say is that blood has higher concentration of glucose, amino acid. Blood has high concentration of glucose, amino acid, right, and of course oxygen. Okay, tissue fluid has high concentration of CO2. Look at it. So there's no red blood cell, no platelets, yeah, fewer proteins, no large proteins. We said less glucose, so it has less glucose compared to the blood. Fewer white blood cells, neutrophils. Okay, lower concentration of carbon dioxide. Okay, more high concentration of carbon dioxide, yes, because it's closer to the tissues. Okay, scientists compare the features of physiology of 3 groups of people who live at different altitudes. These people were selected from populations that have lived at these altitudes for many thousands of years. The scientists took blood samples from people in each location and measured the concentration of hemoglobin in the blood. So they measured the concentration of hemoglobin in the blood, and they measured the percentage saturation of hemoglobin to oxygen in blood leaving the lungs. This is lungs, not lungs. This is lungs, okay? The oxygen concentration in the blood leaving the lungs, okay? Also measured. So this is a table, okay? Okay, let's look at what they want. What do they want us to do? Describe the changes in mean percentage saturation of hemoglobin with oxygen and the mean hemoglobin concentration in blood as altitude increase. So the mean, pause the video and try it. So we're describing the changes in mean percentage saturation of hemoglobin with oxygen. That's mean percent, okay? And the mean hemoglobin concentration in blood as altitude. This is the mean concentration of hemoglobin. Alright, and that. So you can see that as altitude increases from this to this, and that what is happening to mean hemoglobin concentration, it increases, right? Mean percentage saturation of hemoglobin with oxygen decreases. That's it. And you know the reason. They didn't say we should explain, they said we should describe. So just state the trend and quote data. For example, at 100, this was 15.3, but when it increased to 3,500, it became this. So it increased this thing, but this decreased. Okay. So the reason is because as altitude increases, the partial pressure of oxygen in the alveoli decreases. So the affinity of hemoglobin for oxygen decreases. So hemoglobin binds less with oxygen and becomes less saturated with oxygen. Do you get? Alright, so look at it. Tissue fluid. No, this is not the answer to that. Alright, let's go. With reference to table 3, suggests how the people living at high altitude can have an oxygen, alright, concentration. This concentration. in blood leaving the lungs, similar to that of people living at sea level. All right. So, okay, pause the video. How can people living at high altitude still have the same oxygen concentration? Initially, the oxygen concentration is less, and that's what leads to mountain sickness, dizziness, disorientation. But with time, they adapt, they acclimatize to that higher altitude. Why are they able to have the same oxygen concentration? Because usually the body stimulates, releases a hormone called erythropoietin. So erythropoietin is called EPO. Okay, so EPO, what EPO does is that EPO stimulates the production of more red blood cells. Do you get? This is EPO. Erythropoietin. That's erythropoietin. So this erythropoietin stimulates, okay, the production. Sorry, I'm writing it separately, but it's one word. Poietin. Erythropoietin. What a name, guys. This is a hormone that stimulates bone marrow to produce more red blood cell. Okay, so have more red blood cell, therefore more hemoglobin increases in the blood, therefore more oxygen transported. Is that okay? So look at it, guys. So this is what's happening. That's just one. Okay, we have other points: more red blood cells, larger red blood cells, more hemoglobin. Okay, per red blood cell, more alveoli. Yeah, last also increases. Okay, bigger lungs. These guys have bigger lungs. They pump more blood. Okay, have thicker, okay, ventricles. All right, so all of this, so you can see reference to erythropoietin. See the erythropoietin stimulating production of more red blood cells. Okay, more red blood cells through alveoli capillaries per unit time. So this is why they have the same oxygen concentration. Memorize it, guys. All right. Fig 6.1 shows some of the events that occur when a red blood cell flows through a capillary in the lungs. So the results are shown in table 3.1. Okay. State why there are transport proteins in the membrane of red blood cells to allow the movement of hydrogen carbonate ion and chloride ion. Why do we have membranes to allow their movement? This is chloride shift. Okay. This is coming back and this is going on. Okay, this is happening in the alveoli. So why do we allow membrane? Pause the video and try it. Membrane proteins are needed because hydrogen carbonate ion and chloride ions are polar. They are both charged, so they are repelled by the phospholipid bilayer, hydrophobic core. So that is why they need protein, because they will be repelled. Why will they be repelled? Because they are polar. See, ions cannot pass through the phospholipid bilayer. Why? Okay, provide hydrogen passageway for faster diffusion. Okay, so they can pass because they are charged. So that protein provides a hydrophilic pathway, all right, for faster diffusion for the two ions. So act as an ion exchanger. All right, Fig 6.1 shows that chloride ions move out of red blood cell. Explain why this movement is necessary when red blood cells flow through the capillary in the lungs. All right, why does it move out? Pause video and try. Why does it move out? Can you see that hydrogen carbonate ion is moving in, so chloride ion has to move out, so that we don't have excess negative ions in the red blood cell. Okay, to maintain the electroneutrality. All right, to balance the charges, electroneutrality of the red blood cell. All right, so see, look at that. Yeah, see, to balance the charges, maintain electron neutrality, to prevent buildup on the negative ions. Okay, let's calculate. Okay, so why carbon dioxide molecules diffuse from red blood cells into the plasma? Why do they diffuse from the red blood cells into the plasma? Carbon dioxide molecules diffuse from red blood cell, right, into the plasma, okay, to be transported, okay, because they can be transported as a gas, dissolved gas, right? Look at it. Concentration of carbon dioxide is lower. Okay, concentration of carbon dioxide is lower than concentration in red blood cells. Okay, Carbon dioxide diffuses down concentration gradient, so that's why it's leaving. So carbon dioxide can be excreted, to enter the alveolus as part of gas exchange at the alveolar surface. So they're diffusing down concentration gradient to be removed, okay, so that they can be excreted because of gas exchange. Alright, the name of the compound indicated by X. So what is X? Oh, yeah, let's go back to that compound. Yes. So we did not even look at it. Look at it. Where is that X? Yes. X is, look at it. So this binds to this to form this. So X shouldn't be hemoglobin. Okay, yeah, it should be hemoglobinic acid, because it used to have hydrogen with it. When hemoglobin binds to hydrogen, it forms hemoglobinic acid. Alright, so this is hemoglobinic acid, this is oxyhemoglobin. Okay, so it should be hemoglobinic acid. Did you get it? Alright, so that's hemoglobinic acid there. Okay. Alright, they say the sinoatrial node, SAN, and atrioventricular node have an important role in the control of the cardiac cycle. The timing of atria and ventricular system and that's must be controlled so that blood passes through the heart efficiently. Okay, so let's look at this. What is it asking us to do? Fig 1.1 is a summary of blood flow through the right side of the heart during cardiac cycle. 3 boxes are not complete, so how do we fill it? So blood passes into the right atrium. So the blood is passing to the right atrium. Why? Because right atria contract, ventricle relaxes. That's why it's moving. Okay, blood passes into the. Okay, see, blood arrives at the heart and the sphere vena cava and the fibula. Okay, so blood passes into the right atrium. So okay, it moves into the right atrium. All right. Okay, well, see, now from here, blood is forced into the right ventricle. Why is it forced into the right ventricle? What's happening? Because this leads to this. Because atria contract, ventricle relaxing to receive blood, atria contracting to force the blood. Alright, so you can see that blood leaves the right ventricle. What's happening? Atria relaxes, atria diastole or relaxes, ventricular systole contract. So ventricle is contracting here. That's why it's able to, blood is leaving the right ventricle. It's forced out of right ventricle into the pulmonary artery. Okay, so when this is happening, so the atria, you can see when this is done, so the atria starts contracting while ventricle continues to relax. Is that okay? Alright, so I've completed it. It says impulses sent out by SAM pass to the AVN where there's a short delay. With reference to figure 1. Explain why it's important for control of cardiac cycle that there's a short delay at the AVN after impulses have been sent out by SAM. Now it's very important. I wanted to explain that. Why is there a short delay? So that atria and ventricles do not contract at the same time. So that atria empties blood and ventricles filled with blood. Right? Alright, look at it. That's basic stuff there. Atria and ventricles do not contract. Okay? Ventricles are not insist to have. Okay? Alright, they shouldn't contract at the same time. That is why this AVN delays, alright, the passage of electrical impulses to the ventricular wall. Alright, so that's one mark. The changes in blood pressure occurs in heart during the cardiac cycle. These changes cause the opening and closing of bronchial skin. Alright, valves and aortic pulmonary receiving on the valve. Explain how blood pressure changes cause the opening. Now this bicuspid valve, this is tricuspid valve, okay, is the valve that separates the right atrium from the left atrium. Sorry, the right atria from the right ventricle. So why does it open? It opens because the pressure in the right ventricle is lower than pressure in the right atrium. That's why it opens, so that blood can move into the right ventricle. Okay, how does that cause the opening of pulmonary valve? Why does pulmonary valve opens? Because the pressure in the right ventricle is greater than the pressure in the pulmonary vein, pulmonary artery, right? okay, so that blood can pass to the pulmonary artery. Help the flow of blood through the heart. Okay, how does it okay, yeah, how does that help the flow of blood through the heart? Okay, yeah, because the blood, the heart, the valves allow only one, okay, it allows flow of blood in one direction. Do you get? So look at it. Opening of tricuspid, pressure in right ventricle becomes lower than pressure in right. So once it becomes lower, blood can move from right atrium right to the right ventricle. Pressure in right ventricle becomes higher than pressure in right ventricle. Okay, and that was saying it. So opening. Right. So pressure in right atrium becomes higher than pressure in right ventricle. So for opening of pulmonary, pressure in right ventricle becomes higher than pressure in pulmonary artery. That's why it opens. Pressure in pulmonary artery becomes lower than pressure in right ventricle. Okay, so that's why it opens. Help flow of blood right throughout the heart. So the valve ensures one-way flow or prevents backflow, directs flow in correct direction. Okay, blood flows from area of higher pressure to area of low pressure. Is that okay? Yeah, this is very critical, very important. Okay, 3 marks. So thank you for hanging on. All right, so wish you the best for exam today, guys. Make sure that as you go into the exam hall, start with questions you know very well, read it, alright, background information very well and understand it. Do not rush into responding. Try to have an outline for response. Okay, so this is Shedro from that University. I wish you the best. Bye.